All right, so we've already covered mitosis. And the good thing is that if you understand the concepts of mitosis, meiosis is very easy to understand. But there are some important differences between the two. So as we know from just introducing these, what's the difference between mitosis and meiosis? Sebastian?
Mitosis is asexual and meiosis.
Okay, mitosis is used in asexual reproduction. Meiosis is only used in sexual reproduction. Grace?
Mitosis produces like 2 daughter cells. Meiosis has 4.
Yep, the number of daughter cells are produced are different. Mitosis generally results in 2 cells. Meiosis results in 4. Caitlin?
In mitosis, it maintains chromosome number, but in meiosis, it's not.
Yeah, and this is probably the most important difference in that after the process of mitosis, the daughter cells have the same number of chromosomes as the original parent cell. It maintains chromosome number. Meiosis, however, is sometimes called reduction division. It takes the number of chromosomes in the original parent cell, and then the daughter cells that are produced actually have half the number of chromosomes as the original. So it reduces the number of chromosomes in half. So meiosis, okay, sometimes called reduction division, takes and splits the number of chromosomes in half. And when would living organisms need cells with half the number of chromosomes? Yeah, to produce gametes. That's what meiosis does. It produces gametes, sex cells. Takes diploid cells. Why don't we introduce those terms? Diploid cells have the full chromosome number. For example, in humans, diploid cells have 46 chromosomes. And it produces haploid cells, cells with half the number of chromosomes. In humans, that would be 23. We call this process gametogenesis, the production of gametes. It's used to produce sperm cells and egg cells, to produce the reproductive cells in plants as well. This happens through this process of meiosis. And in meiosis, there's actually 2 divisions. The cell splits 2 times. Okay, an initial time and then a second time. And that's why it produces 4 cells. Because you have 1 cell that produces 2 cells, and then they each split, forming 4 cells. The phases are called the same things. So it's still interphase, prophase, metaphase, anaphase, and telophase. The same general events happen in each of the phases, but there are some slight differences in the first division. Okay? So often we split these, call it meiosis 1 is the first division, and meiosis 2 is the second time it divides. So an interphase, okay, before a cell is going to go through meiosis, the same sort of things happen. It's the resting stage. It's between divisions. Okay? During interphase, the chromosomes replicate. The DNA makes an identical copy of itself. That's the same as mitosis. Corbin?
What cells are turned into sperm cells, like your egg cells?
Sort of precursor cells. They're cells in the ovaries, cells in the testes, that are sort of the precursors of sperm and egg cells. So they come from other types of cells in females. Those initial cells that are going to become egg cells are present. They sort of start to divide, and then they're sort of frozen in prophase of meiosis 1 in the ovaries from about 20 weeks into development. So during the fetus, in a female fetus, the embryos start meiosis and they kind of get frozen in prophase 1 until puberty. And after puberty, when ovulation begins, once each reproductive cycle, one of those cells finishes out meiosis and produces the actual haploid egg cell that can be fertilized. We'll talk a little bit more about that towards the end here. So the phases of meiosis, with prophase 1, metaphase 1, anaphase 1, telophase 1. And probably the most important key difference for you to recognize happens in these first two phases. We know that in prophase, I said remember P for prepare. The nuclear membrane starts to break down. Okay, the centrioles form, spindle fibers start to form. The chromatin condenses to form chromosomes. Okay? And during this phase, something called synapses takes place. What happens is that homologous chromosomes sort of pair up together. They match up. And they form something that we call a tetrad. This is a tetrad. It's two homologous chromosomes that have lined up with each other. So let's go back to this homologous chromosome. What is it? So I guess let's go one step further. Here we have two sister chromatids that are forming a single chromosome. How do these two chromatids compare to each other?
They're identical.
They're identical. They're copies. When we said the DNA copies itself during interphase, that's what happens. One chromatid is copied to form the other identical chromatid. So these two things are exactly the same. Then we have these homologous chromosomes. Okay? They're similar to each other, but they are not identical. Okay? Homologous chromosomes contain the same kind of information. The chromosomes are the same size. Okay? The centromere is in the same place. But they're not identical. They have genes for the same sort of traits. For example, if there's a gene for eye color up here on this chromosome, there's also a gene for eye color on its homologous chromosome. But this one may be a gene for brown eyes. This one may be a gene for blue eyes. One of these homologous chromosomes came from who? Each parent. One from the mother, one from the father. So we inherit our chromosomes in pairs. During meiosis, these chromosomes line up back with their homologous partner, and they form this thing that we call a tetrad. So two chromosomes line up, and we call that process synapses. Now, sometimes when they line up next to each other, these homologous chromosomes, a process called crossing over can happen, and that's what's being shown here. Okay, we have our synapses, we have our two homologous chromosomes lined up next to each other. But sometimes they sort of can cross and exchange pieces of the chromosome. So here, this blue chromosome and this red homologous chromosome, they kind of swapped, and a piece of one chromosome can be transferred onto the other. And so we see that, okay, that process here. This is one of the reasons that sexual reproduction can lead to an increase in variation in a species. Because you take these chromosomes, you're actually sort of mixing and matching pieces from two different homologous chromosomes, and that increases the variation. So that process is called crossing over. Why can't it happen in mitosis?
Because there's no variation.
Well, but if this happened in mitosis, there would be variation. What's that? They don't line up like this in mitosis. How do they line up in mitosis? Single file, individual chromosomes straight down the middle. It's only in meiosis where these chromosomes align, form these tetrads during synapses, and then can cross over.
Do they have to cross over?
Nope, sometimes they do, sometimes they don't. It's just sort of a random process.
So that's sort of like saying that's maybe why you have this trait from your father.
Yeah, it could be that this chromosome you inherited from your mother, it doesn't necessarily stay completely intact in all of the other cells that are produced in meiosis. It could then get some of your father's chromosomes. So you have the sort of shuffling of the deck.
Oh, is that why, like, sometimes both of your parents are, like, brown hair, but you get different?
It could be. It could be because of the crossing over, or because of which genes, or it could be because of dominance. So there's a lot of different factors that come into play there. So in prophase, synapses happens. In metaphase, the spindles attach. Okay, again, just like it did in mitosis. The spindles attach to the chromosomes, which have aligned up with their partners on the equator of the cell. What do you think happens in anaphase? They get pulled apart, but now the two sister chromatids do not get pulled apart. Each of the homologous partners get pulled apart. Okay? So the spindle fibers pull the homologous chromosomes and separate them from their homologous partner. Pull them to the poles of the cell.
So they exchange and then they pull apart.
Yep, so the crossing over can happen and then they get separated. And then no more crossing over because they're no longer in tetrads. And then in telophase, what do you think happens?
So
Cell separates, splits. The cell splits in two. New nuclear membrane forms, just like in mitosis. But now we have two cells. How many chromosomes are in those cells? Will they still have, in humans, 46?
Yes.
No, only 23.
23.
Because those homologous partners lined up, 23 homologous partners. When they each partner gets split up to go to opposite poles, then those resulting cells only have 23.
Won't those each have 46?
They have 46 chromatids, but only 23 chromosomes, right? Because this one's going this way, this one's going this way. Let's look at it here. So this is the first division. Again, interphase, in-between phase. Chromosomes duplicate, okay? Prophase, chromatin condenses to form these chromosomes. We have our tetrads have formed, okay? Crossing over may occur, okay? They move to the equator of the cell, but now they're with their partners. Okay? They're along the equator. The tetrads are aligned. They get pulled apart. See here, crossing over did occur. Chromosomes get pulled to opposite ends of the cell, and then the cell is going to split. Okay? So here, okay, we have the same thing. This is showing us the whole meiosis. So again, chromosomes duplicated. The chromatids duplicated form these chromosomes. They line up with their partners. They get pulled to opposite sides, and we result in two cells. Okay? From here on out, these two cells are going to split again. The rest of the stages, meiosis 2, is basically the same as mitosis. Okay? So again, the daughter cells. From meiosis 1, they each now have half the number of chromosomes because those homologous pairs split up. What happens in metaphase? What do you think happens in metaphase? They line in the middle. Those chromosomes line in the middle of the cell, along the equator. What happens in anaphase? The centromere divides. The two sister chromatids start to separate from each other. They start moving to the poles.
why is it sister and not brother?
I don't know.
Because they're sexist.
No, actually, I think it's like the opposite. Because if it was brother, that would be sexist.
I don't know.
Oh, whoa, that's sexist. That's sexist.
Wow. Eventually, those sister chromatids get pulled to opposite poles.
They just think girls are superior, so they're just.
All right. And then what happens in telophase?
Sisters and daughters, not brothers and sons.
Antonio, they split apart. Again, the chromatids separated. They get split apart. New nuclear membrane forms, and our end result is 4 cells. But They're haploid cells. If we look in this diagram, our starting cell, how many chromosomes did it have? No, in this cell that's shown here. No, and no. 4 chromosomes. 1, 2, 3, 4. Our resulting cells, 2 chromosomes in each. The number of chromosomes got reduced in half. 1 cell produced 4 cells. That's meiosis.
Wait, starting with the first one, why? So there's one big blue, one little blue, one big red, one
Yep.
little red. If you put them together, that would only be 4, but.
But they don't, so this is before they've doubled.
Oh, okay.
So this one comes when this copies its other half.
Okay.
So the X chromosome, they're exact same on either side.
2 sister chromatids are
They're
identical.
considered one.
Yes. Again, this is just a sort of another view. Again, this is meiosis 2, the second division. Okay, our chromosomes, which have split from their homologous partner, okay, to form these 2 cells. We see crossing over has occurred. Okay, they align in the equator. The 2 halves of the chromosome get pulled apart in anaphase. They move to opposite ends of the cell. New cells form, and again, number of chromosomes reduced in half. Nate?
Wait, so does any crossing over or synapses occur during meiosis too?
No, who could tell me why?
Just they're lined up.
Because they're lined up in the.
Not, but why are they lined up single file?
Because they got separated.
They don't have a homologous partner anymore. Because where did its homologous partner go?
The other cell.
It's in this other cell now. So they don't have partners anymore. There's just one of each pair. Yep, Cormac?
So meiosis 2 is pretty similar to mitosis.
It's basically mitosis. It is mitosis. Yep. Meiosis 1 is the difference. In meiosis 1, really, the main difference is that the chromosomes line up with their homologous partner, which gets split up, rather than lining up single file. All right. So you know about meiosis now?
Yeah.
Tomorrow we'll talk a little bit about development, and then we'll have covered what we need to cover for the next day. So you do have workbook pages due tomorrow.